Welcome to the problem-solving set on transcription, RNA, and the central dogma. Question 1. The coding sequence for gene 1 is read from right to left on the figure, whereas the coding sequence for gene 2 is read from left to right. Determine which strand of DNA, either the top or the bottom, serves as the coding strand in each gene. Well, there are some things about RNA polymerase that you have to remember in order to answer this question. So like DNA polymerase that we encountered when we did DNA replication, RNA polymerase reads the template strand in the 3' to 5' direction, and it makes the new RNA strand in the 5' to 3' direction. Okay, so for gene 1, it's saying that the coding sequence is read from right to left. So it's read this way. So it's asking which is the coding sequence strand, and then we could also answer which is the template strand. Well, the template strand is going to be the one that's read, and that's going to be the 3' to 5' direction. So for gene 1, the top strand is 3' to 5', right, when you're going from right to left. So the top strand is the template strand, and therefore the bottom strand is the coding strand. And the coding strand is the one where the DNA sequence is very similar to the RNA sequence that they're making. The only difference is that the thymines are changed to uracils, right? And that's the difference between RNA and DNA in terms of the bases. So for gene 1, the top strand is the template and the bottom is the coding. Now for gene 2, the coding sequence is read from left to right. So which one would be the template here? Remember that the template is the one that runs from 3 to 5. So if you're going from left to right, the bottom strand is going from 3' to 5'. So for gene 2, the bottom strand is the template, and that means that the top strand is the coding strand, or the one that's analogous to the RNA sequence being made, except for the switching of thymine and uracil. Okay, so that is question 1. Question 2. Consider in the following statements about transcription and determine which are true. If false, correct to make true. Statement A. Downstream from the promoter, the coding strand of a gene runs 3' to 5'. So remember, we just did this in the last question, that the coding strand of the gene is the one that actually runs 5' to 3'. So this is false, and coding runs 5' to 3'. And the template strand is the one that runs 3' to 5'. And that's because the enzyme RNA polymerase reads the template strand 3' to 5' in order to make the new strand in the 5' to 3' direction. Okay, statement B. Structural genes produce mRNAs through transcription and eventually a polypeptide. So that is true. That is the sequence of transcription and translation. The genes produce mRNAs, messenger RNAs, through transcription, and then eventually those mRNAs are converted into polypeptides through translation. Statement C. A site in the DNA sequence designated +20 would be located within the promoter region. Now this is false because of the +20. So remember that promoters are actually upstream of the coding sequence. So promoters are upstream of coding. And the coding region starts at +1. So anything that is upstream of this coding sequence +1 would actually be negative. So I'm going to write that as negative, and that the coding sequence starts at +1. So the site in the DNA sequence that's designated as +20 would actually be downstream of the coding sequence and would be part of the coding sequence. Statement D. The RNA polymerase holoenzyme found in bacterial cells contain 4 subunits. So this is a pretty tricky one, but it actually is false, and I'll tell you why. The core enzyme of RNA polymerase does in fact contain 4 units, but in order to be considered the holoenzyme, it needs to include that sigma factor that we talked about in the theory session. And so that actually equals 5 subunits. So we'll say that RNA pol has 4 subunits, and that's the core of the enzyme core. But I'll write it down here. The holoenzyme is equal to the core plus the sigma factor, right? So that is a total of the 4 subunits in the core enzyme plus another subunit that's the sigma, so a total of 5 subunits to get that holoenzyme. Statement E. Both proteins and some RNA molecules can be biological catalysts. So this is true. So that's the end of problem 2. Problem 3. For each of the following RNA molecules, determine the double-stranded DNA sequence it was transcribed from. Be sure to label the polarities of each strand and label the template and coding strands. So remember that RNA and DNA are pretty similar, but they do have some distinct differences. They have a different sugar where DNA has deoxyribose and RNA has just ribose, so it has that extra hydroxyl group. And they also use different bases, so they both use guanine, cytosine, and adenine. But in RNA, instead of using thymine, they actually use uracil, which is labeled as a U. So we're going to do, for each of these, we're going to find the double-stranded DNA sequence it was transcribed from, and then we're going to determine which strand was the template and which was the coding. So let's look at A. Remember that the coding strand in the DNA is exactly the same as the RNA that was made, except that the uracils are changed for thymines. So in order to get the coding sequence, we're just going to copy this, so it's going to be 5' G, C, A. Now here, the U, uracil in RNA, is going to be a thymine in DNA, so we'll write a T. Then it's going to be G, C, A, 3'. So this was the coding strand. Okay? And antiparallel, the other part of this double helix in DNA, is going to run 3', C, G, T, T, A, C, G, T, T, 5'. So this was the template strand. And just to be complete, we want to write that transcription happened from left to right. Because remember, the template strand is read from 3' to 5'. So I'll write transcription here. Okay, for B, again, to get the coding strand, you're just going to copy the RNA sequence that you're given, but change out the uracils for thymines. So remember to keep the polarities as the same as well. So for B, the coding strand is going to be 3' here, T, 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 G, C, A, T, 5'. So this is the coding. And the template is going to be the antiparallel match. So it's going to be 5', A, C, G, T, T, A, 3'. And this was the template strand. Now remember again, the template strands are read from 3' to 5' by RNA polymerase, which means that transcription actually happened from right to left for this sequence. And finally for C, to get the coding strand, it's going to be 5' G, T, remember the U's are changed to T for DNA, A, T, C, G, C, A. So this is coding. And then just find the complementary base pair for template. So C, A, T, T, A, G, C, G, T, 5'. So that's template. And again, template's read from 3' to 5' by RNA polymerase, so transcription happened from left to right for sequence C.